Good day, everyone. My name is Joy Concepcion from QPS, and I will be the moderator for today's webinar. On behalf of QPS, I'd like to welcome you to the Anti-Drug Antibody Analysis for Preclinical Studies. This webinar will discuss the different considerations for preclinical and clinical assays, as well as the current QPS fit-for-purpose approach for the ADA assay validation and bioanalysis of samples from preclinical studies. It will be presented by Dr. Monique Putman. A few housekeeping comments before we begin. Everyone will be muted during the presentation. Please enter any questions under the control panel. If we cannot get to your question, we will respond to you directly after the webinar. This webinar will be available on demand to all registrants. We will send you an email once the recording is available. And now I'd like to introduce our presenter, Dr. Monique Putman. Dr. Monique Putman obtained her PhD in microbiology at the University of Groningen. She has been at QPS since 2002 and is currently the Director of Translational Medicine at QPS Netherlands, where she leads PK and immunogenicity groups in the method development, validation, and support for discovery, non-clinical, and clinical studies of protein biotherapeutics using ligand binding methods. And with that, I would like to welcome Monique.
Hello, everybody. Good afternoon, and welcome to our webinar on anti-drug antibodies, specifically on the current approach towards antibody analysis for preclinical studies. ADA analysis is typically important for biological drugs. Biological drugs are an increasing group of the drugs that are in development or approved by regulatory agencies. And these are peptides, monoclonal antibodies, other proteins or oligonucleotides. And these typically are very complex molecules that can be recognized by the immune system as a foreign molecule, triggering the generation of antibodies. This drug-induced immunogenicity is recognized as a major challenge in the development of biological drugs. The immune response, or the antibodies against the drug, can be triggered by two different pathways. In the first pathways, there are specific antigen-presenting cells that take up the protein that is recognized as foreign and present a small portion to the other cells in the immune system. And this portion can be 6 to 9 amino acids approximately, so it's only a small fragment to which the antibodies are directed. In the end, it triggers an entire cascade of reactions in the immune system, leading to the generation of a plasma cell that in turn will create the antibodies. The second pathway is a B-cell dependent pathway. This occurs if there are multiple epitopes on a molecule, so they're repeating structures or aggregates that can cross-link different receptors on a B cell, therefore by triggering it to develop into a plasma cell and creating antibodies. There are several risk factors in the immunogenicity. These can be a genetic background of a patient. This, for example, determines how the antigen-presenting cells present the antigen to the immune system. The disease state can be of influence. For example, in cancer treatments, early on in the cancer, there can be quite some immunogenicity, while in later stages, the immunogenicity goes down. And co-treatment, for example, medication that suppress the immune system, also affects the immunogenicity. Other factors that affect the immunogenicity of the drug are related to the drug itself. It can be the dosing, the frequency, the route of administration. For example, intravenous injection is generally more immunogenic than subcutaneous dosing. And can also be particular things for the drug itself, so the the production process that leads to particular post-translational modification, the formulation, any impurities that can be in the product, as well as the origin of the molecule. An example of this is monoclonal antibodies. So to reduce the immunogenicity of monoclonal antibodies, common approach is to reduce the amount of murine sequence in a molecule and make it look as much as human as possible. And in this sense, there are different types of molecules, completely murine molecules, molecules where the constant part of the molecule is replaced by human sequences. This is called a chimeric molecule. A humanized antibody is a molecule where also part of the variable regions are replaced by human sequences. And then finally, there's the completely human molecule where also the antigen binding sites are replaced by human sequences. And the further you go to the right in this picture, the lower the immunogenicity that is expected in human. However, if you dose these drugs to animals, there can be an increasing amount of ADA. So what is the problem with this ADA? Well, first of all, it's a safety concern. The presence of ADA can lead to hypersensitivity reactions, and it can also affect the clearance of the drug. In many cases, the binding of antibodies will decrease the clearance and increase the clearance, and the exposure to the drug is reduced. However, there have also been some examples where the antibody binding decreases the clearance, leading to a higher levels than expected. And in case of multiple dosing, that can even lead to overdosing. In addition to the binding antibodies, which gave these points, there can also be neutralizing antibodies. And in this case, the antibodies bind to the active site of the drug, thereby inactivating it, causing a loss of action. And in some worst cases, even can inactivate the endogenous counterpart, making the disease worse rather than better. In order to follow these formation of ADA assays, an assay needs to be set up. And this is typically a ligand binding assay. In ligand binding assays, the assay is depending on the interaction between the antibody and the antigen. Typically, samples are diluted and captured on the plate. In an ADA assay, the capture reagent is the drug. And then the sample with potential ADA is added to the well and can bind to the drug. Then a suitable labeled detection reagent, which can either be a labeled drug or conjugate that is specific for the antibody of the intended species. And then a suitable substrate is added that can cause a colorimetric readout, a fluorescent signal, or a luminescent signal, which can then be measured by suitable equipment. The most used assay for ADA analysis is a bridging antibody assay. And this is depicted in this figure. The labeled antibody, or actually two versions of the labeled antibody, is incubated with the sample. And then the present anti-drug antibodies can link the two labeled versions of the drug, which can then be captured on a plate, and the signal can be introduced. In many cases, it is performed on an MSD format. The advantage of an MSD plate is that it does not require a lot of washing, which also allows the detection of low affinity antibodies. Further, in a bridging antibody assay, the assay is independent of the species in which the positive control antibody is generated. However, in some cases, you see that the drug can form dimers or multimers in the absence of anti-drug antibodies. And in this case, such a bridging assay format is not suitable. In that assay, in that case, an indirect ADA assay format can be used. In this format, the plate is coated with the drug. The antibody present in the sample can bind to the drug, and then a suitable detection antibody that is usually specific for the species of interest, or can also be a protein AG labeled version, and then develop a signal. An important part of the ADA assay is the positive control antibody. And this is meant to represent the polyclonal antibody response in the study samples. And it is used to estimate the sensitivity, precision, and drug tolerance during the validation, as well as the performance of the assay during routine analysis. As the immune response in study samples will be polyclonal, it is preferred also to use a positive control antibody that is polyclonal. This can be obtained by hyperimmunization of animals and then subsequent purification of the antibodies. In some cases, it is possible to obtain a positive control antibody from a patient matrix, from a suitable patient population. However, not in all cases it's possible to obtain a polyclonal antibody response, and in that case, a monoclonal antibody can be used instead. In contrast to a PK assay, an ADA assay typically does not have a calibration curve. Instead, a cut point is set, which is a threshold below which samples are considered negative for the presence of ADA, and above which samples are considered positive for the presence of drug-specific antibodies. Typically, this cut point is determined by measuring a series of serum or plasma samples from drug-naive patients from a suitable population, and by statistical means, a cut point is calculated. In ligand binding assays, there's quite often some variability in the signal between runs. And in order to correct this cut point value for this variability between runs, often a floating cut point is used. In that case, cut point factor is established during the validation, and the plate-specific cut point is calculated based on the negative control that is included in each plate and the cut point factor. If we look at the ADA analysis in clinical studies, the primary focus is patient safety. As indicated earlier, it can trigger hypersensitivity reactions and also give issues with the exposure to the drug. And therefore, there's a lot of attention to make the assay as sensitive as possible. The sensitivity that's required for human ADA analysis is 100 nanogram per ml as described in the guidelines. And the cut point is set such that there's a 5% false positive rate in the screening assay. And this is done in order not to miss any low positive responders. The preference is rather to have a little false positive rate and be on the safe side. The low positive control assay that is used in results validation as bioanalysis is set to a level just above the cut point to ensure that in every run the sensitivity remains more or less the same, and it is set in a statistical way to at a level that gives a 1% rejection rate. So in 1% of the bioanalytical runs, the low positive control will be below the cut point. So the analysis is done in a tiered approach, which means there's several steps to be done. The first step is a screening assay. So the sample is analyzed, and using the cut point as determined in the validation, it is judged whether the sample is negative or positive. In case the sample is positive, it is tested whether the response that is found is drug specific. And this is typically done by adding an excess of drug, which will inhibit the assay signal in most assays. If you see a reduced signal in the presence of excess drug, the sample is confirmed to have drug-specific ADA. If the response is a non-specific background, it will not be affected by the presence of excess drug, and the sample is then confirmed as not confirmed. A third step is a titration assay. This is performed for the confirmed positive samples, and it gives you a sense for the amount of presence, antibodies present in the sample. Most often, the sample is measured at several dilutions, and the highest dilution at which it is still above the screening cut point or titration cut point is reported as the titer. In addition, for the confirmed positive samples, it can be tested whether these are neutralizing or not, or further characterization can be performed to verify what the antibody is directed against or what the type of antibody response is. In preclinical species, the focus is different. In this case, the primary focus is the explanation for any potential deviating PK curves. As you can see in the picture presented here, the presence of ADA can trigger a faster clearance of the drug, giving a much lower PK results. And these would need to be explained. Mostly samples are collected and banked, but only analyzed if needed. And important to realize that the ADA in preclinical species is not predictive for the immunogenicity in human. I'm sorry. Given the higher dosing in preclinical studies, the drug tolerance is more important than the sensitivity. The tolerance is the amount of drug that can be tolerated without giving a negative signal for a positive sample. And in the method development, there's much more attention to the high levels in the preclinical study. And initially, there was not a lot of guidance on the ADA validation. And there were some white papers published that described the ADA validation for both clinical and preclinical species. And the approach was more or less the same for both. When the FDA and EMA guidances came out, it became clear that the ADA analysis for preclinical species could be approached a little bit differently. It is indicated that in the EMA guidance is indicated that the ADA assay should be validated, although it's not specifically indicated how you should do that. Whereas in the FDA guidance, it is indicated that the guidance is intended for the analysis of clinical samples. So this triggered quite some discussion on how to proceed with the analysis of preclinical samples. So first I will take you through the approach that we followed, I think, up till 4 or 5 years ago, in which the ADA sample of ADA assay validation for human and preclinical species was approached in a very similar way. For the screening cut point determination, we would typically measure a set of individual samples in 6 runs by 2 technicians on 3 days in a balanced design. For preclinical species, the number of individuals would be lower than for human, but otherwise it would be similar. It would be evaluated in the 6 runs whether the variances were equal, which allows the use of a floating cut point. And then a statistical calculation of the screening cut point was performed based on a 5% false positive rate. In addition to the screening cut point, also a confirmatory cut point was calculated. In this case, all the individual samples are measured again in the 6 runs with and without excess of drug. And then the signal inhibition in the presence of excess drug is calculated. This typically varies between -20 and +20% due to variability in the assay. And then the cut point was defined based on a 1% false positive rate. In a subsequent step, the sensitivity of the assay is established. This is done by diluting the positive control and measuring 6 or at least 6 dilution series of this positive control, and calculate the intersection point of the fitted dilution curve and the cut point. And from that, the low positive control could be calculated. Another part that was standard in the validation is the selectivity. There are some matrix components that can inhibit the ADA from binding to the capture and detection reagents and give different results. And in order to avoid false negative results, it's evaluated whether the assay is suitable. For that, 10 individual samples were measured, both spiked and at a low positive control level, and the unspiked samples should be below the cut point and the spiked samples above the cut point. In addition, the effects of hemolysis and the high lipid content is evaluated. With regards to the acceptance criteria, we evaluate the amount of positive and negative samples and do not calculate a recovery because we consider that not relevant. The next part in the validation is the precision. For that, both intra-run and inter-run precision is measured, either 5 measurements in 1 run or 2 to 3 measurements in 6 runs. And that is performed for both the screening and the confirmatory assay, and the CV had to be below 20. And finally, the stability is assessed, which is, in general, antibodies are very stable molecules. And you also have to bear in mind that the study samples contain positive control, are not the same as the positive control antibody. So you can establish the stability of your positive control, which can be like a monoclonal or rabbit antibody in the human matrix, and it will tell you how you can handle the positive control antibodies, but not so much the stability of the study samples. Over time, there was quite some discussion on all the experiments and whether or not it could be reduced for preclinical species. And I will take you through the current approach at QPS towards the validation of ADA assays for preclinical species. This can be applied for all the different species that can be used before entering into phase 1 studies. In general, only a screening assay is performed. Further, the sensitivity of 1000 nanogram per ml is considered adequate. And the advantage of the reduced approach is that you would need less volume to perform the validation of the assay. In this particular example, we measured 60 measurements of so the cut point is based on 60 measurements of an individual sample. And in this example, we had 2 runs with 30 individual samples. In this particular example, we measured the samples in duplicate wells. And you can see that 3 of the samples had a high duplicate well CV. If during method development it is observed that the duplicate well CV is quite low, you can even consider measuring the samples in single well mode to further reduce the volume that is needed. The cut point is then based on these 60 measurements and 1% false positive rate. In this particular example, the signal to noise ratio was calculated for each of the individual samples by dividing the response of the sample by the response of the negative control. And we have evaluated the distribution. As you can see here, there's a Shapiro-Wilk test performed, and this data was nicely shown normal distribution. In that way, the cut point was calculated by statistical means, very similar to done during human validations. If the data is not normally distributed, the cut point can be calculated using the 99th percentile of the data. The next step is the sensitivity. For that purpose, dilution series of the positive control are measured, and in this case, it is limited to only 4 dilution series. And the low positive control is not based on a 1% failure rate, but is set at a reasonable sensitivity, which is equal or lower than 1000 nanograms per ml. In this particular example, we measured 4 dilution series, and the mean concentration that corresponded with the cut point was 26 nanograms per ml. And if calculated with the 1% failure rate, the LPC level would be set to 92 nanograms per ml. But in fact, in this validation, it was set to 500 nanograms per ml, as that was considered sufficient. In subsequent step, the precision was evaluated both at a low and high positive control level. It was done in 4 runs, which shows a nice precision, and the drug tolerance was evaluated and passed. There are some optional validation parameters that can be selected if needed. These include the selectivity. Typically, selectivity is considered not required, and the animals used in the preclinical species are often inbred lines and do not vary a lot. However, if wanted, you can evaluate the selectivity. Further titration is usually not performed. In case the drug tolerance is found to be suboptimal, you can consider measuring the samples at several dilutions to verify if the result does not change and if the amount of drug is diluted out. And finally, the stability can be assessed if you want to have more certainty about how to handle the positive control samples. So overall, if we compare the 2 different validation approaches that we have applied in the past and now, you can see that the number of runs is seriously different. And especially the amount of matrix used is different. So in both cases, the screening cut point is used, but the number of individual samples is reduced from 180. This means that was 6 runs with 30 individuals to 60 measurements. There's no confirmatory cut point established. The sensitivity is done with 4 series rather than 6 series. The low positive control is not calculated on 1% rejection, but set at a reasonably low level. The selectivity can be skipped as well as the stability, but the other parameters should be evaluated, such as drug tolerance, the boson effect, and the precision. So with this, I would like to complete our webinar and would be happy to answer your questions.
Thank you, Monique. And now we will begin the question and answer portion of the webinar. Please enter any questions under the control panel. Our first question, when people do PK for biosimilars, off-the-shelf assays are standard. Is that the case for ADA assays in support of biosimilars? Can we just use the assay that you've already prepared?
No, in ADA assay, actually you should use your own drug as a reagent, so for capture and detection, because there may be some impurities or other things that may be different from the originator drug that you need to pick up. Of course, if there's an assay available for the innovator drug, you can use the positive control or the assay conditions, often also for the biosimilar. So it's not a complete new assay, but there needs to be a new validation.
Great. What is the difference between the format of developing ADA assays for monoclonal antibodies versus multi-specific biotherapeutics?
In the format of the assay is typically the same, so you follow the same approach. However, if there's a molecule which has different moieties, you would like to know to which moiety the antibodies are directed. So in that case, you can use a confirmatory step to look at yeah, what the ADA response is directed against. And if you, for example, do a bispecific antibody, you can do a confirmatory step with each of the arms of the antibody and check where the antibodies are directed against.
Great. In the development of pegylated drugs, is there any differences between ADA assays for pegylated drugs versus monoclonal antibodies?
Well, I think again the assay format is typically the same. However, there's in human quite some pre-existing antibodies against PEG, so it is more difficult to set a cut point, and you should probably screen quite some samples to have a good set of samples to do the cut point calculation. So that is more the difficult part.
In case ADAs have been shown to be non-neutralizing in one species, is it automatically the case for other species, such as like non-rodents? So if you saw it in mouse, will you see it in like dogs or monkeys?
Not necessarily. I think for every species that can be different. Of course, as I briefly went into the immune system, that works differently for each of the species. So it could very well be that in some species there are neutralizing antibodies, whereas in others are not. So I would not exclude it just because it was not observed in one species that it will not happen in other species.
Okay, great. And then how about ADA assays against antisense and mRNAs? Is it different?
It's a little bit different. Initially, there was not a lot of interest for ADA assays against oligonucleotide drugs because nucleotides are not so immunogenic by nature. However, currently there are quite some modification in the bases used, and to make it more stable, for example, or modify the lifetime of the molecule. And these modified bases may trigger the immune system to generate antibodies. So ADA is more and more performed for nucleotide drugs. However, it may be quite difficult to get positive control antibodies for these molecules.
Okay. And then how how do you find a positive control when doing ADAs for oligos?
Yeah, so in some cases it has been has worked to immunize hyperimmunize animals with oligos, but that's the low chances for antibodies generated are low because these molecules are not so immunogenic by nature. I've also heard that there have been monoclonal antibodies used towards that.
Right. This will close our Q&A portion of the webinar. QPS would like to thank Monique for her detailed presentation on the consideration for ADA assays in preclinical studies. I'd like to thank everyone for their time and attention today. If we didn't get to your question, we will get back to you after this webinar. You will receive an email when the recording is available for this webinar, and have a wonderful rest of your day. Thank you.